Okay, thank you very much. Thanks to Thermo Fisher for inviting me to speak. I'll try not to refer to AFI. It's old habits die hard in this area, but yeah, we'll see how I go. We're gonna move from four-legged beasts to zero-legged, and we're going to talk about fish. So, yes, Atlantic salmon. Many of you will recognise Atlantic salmon from perspective of the dinner plate as a pretty tasty product. It can be enjoyed in many different ways. Obviously, for myself, working with farming salmon and breeding salmon, we are, of course, looking at it from the previous step before it ends up on your plate. I thought I'd spend a little bit of time just introducing salmon farming, marine harvest, who we are, what we do, why we run a breeding program. Because I guess the majority of the audience here is probably coming from the livestock area, and maybe farming Atlantic salmon is not something everyone knows too much about. So to put things in context a little bit, salmon farming, well, we are kind of the minority here, the salmon people, but I think we can be kind of justifiably proud of our species in a way. It's not as big as of course, the livestock species in terms of production, but still there's a significant amount of production of farm salmon now in the world. It's a very effective species in a way to farm. The yield is around 68–70% in terms of how much edible product you get off the total body weight. Compares very well to livestock species. And I think the one that is most significant in terms of farming of a species like salmon is the feed conversion ratio, which is very good. These figures here are 1.3. I've seen it even lower than that, 1.2, 1.1 even. So it's a very efficient species to farm in many ways. And we work, of course, farming salmon within the natural biology of the species, and the natural biology of the species of salmon is a little bit complex. The fish, of course, start off in freshwater and they hatch in rivers, and then the fish, after a period of time, go through a process of adaptation called smoltification, where they prepare themselves to go out to sea in saltwater. They move out to sea, they grow for a period of time in sea that can range from 1 year to many, many years, and then eventually they return as adults to spawn back in the rivers again. And of course, when we farm salmon, we have to mimic this natural process. So in terms of farming salmon, well, how do we do it? Well, you have an incubator where you incubate the eggs, and this, on a big scale, can be a highly efficient process. Those eggs are then hatched, and these fingerlings are reared for a period of time in freshwater. They are then go through a process of smoltification that we mimic. We can use various treatments to light regimes and adjustment of water parameters to go get this process happening, smoltification, so they're ready for salt water. Then they're transferred to sea. They are grown out to sea up to a slaughter size that can be anything from 4 to 6 kilos, typically. And at that point, they're harvested and processed and end up on your dinner plate. Now, Marine Harvest is a company that has many brands. Maybe some of you are familiar with some of these brands of the product. It's not just Atlantic salmon. Marine Harvest is also involved in some other species and product distribution of other species, but primarily it's salmon. And when I talk about breeding work and genomics, I'm only talking about salmon. Marine Harvest is the biggest producer of Atlantic salmon in the world. It's represented across the world. Of course, we have markets across the entire world. There's a very large turnover, and the harvest volume in 2016 was nearly 400,000 tonnes of salmon. So that's a significant amount of fish being produced now. So why is a breeding program important to Marine Harvest? We're a production company. We produce salmon and sell salmon. So why do we need to run a breeding program? Why don't we just buy eggs and grow up salmon? Well, what we know is we're fortunate in that basically all the important economic traits in Atlantic salmon have a significant genetic component. And these economic traits, well, I won't mention them all, but obvious traits like growth rate, very important, faster growth, less time at sea, more efficient, feed conversion ratio, the quality of the product, good colour, good flesh quality. Disease resistance is now a very important trait. There are many challenges out in the sea, many pathogens, parasites, and these are all very heritable, we see in our data, so we can do a lot with this in a breeding program. Having a breeding program for us means that we can access a stock of fish that they're suited to our specific needs. So we can tailor the breeding program to what we want as a fish to produce in our farms. And our farms have certain challenges, and we want to produce fish to meet those challenges. It gives us control of the whole production cycle from egg to harvest. We also produce the feed, so we're a full production company in it. And having that full control of egg to harvest is advantageous in many ways, we see as a company. And of course, there's also the marketing side. We have all these products, you see all these brands, but actually our breeding program is part of that brand. And maybe some of you have heard of Maui salmon. That's our strain, actually. The Marine Harvest strain is the Maui, excuse me, the Maui strain, and that has its origins back in the 1960s. So Marine Harvest has breeding programs across the world, mainly three main really sites for when it comes to selective breeding work. There's the Maui population in Norway. There are three to four strains in Chile that we farm and run a breeding program for, and we have 2 strains in Canada that we have in a local breeding program run in Canada. And yes, I won't spend much time on this, but this just shows that within Europe we also can supply eggs to Scotland and the Faroe Islands from the Norwegian breeding program, and Chile is only producing eggs and fish for Chile, and likewise Canada is exclusively for Canada. So what do we breed for? I mentioned some of the important economic traits before, growth rate being one of the most obvious, most important traits, and the one that's been included in the breeding programs for the longest time. Of course, you remember since this began in the 60s, there was no genomics back then. Breeding was much simpler, and growth was the natural trait to select for. As I mentioned, also quality traits. Here's the flesh colour. Pink or red flesh is an important phenotypic quality for salmon. Some markets especially highly value this, and it's a heritable trait too. This is 2 fish that have been suffering from a viral disease. So disease resistance not only kills fish, but it also results in significantly poorer quality. These fish are effectively runts now. They've stopped growing, and you can see the condition is quite terrible. And another major problem that the industry has is this little critter that's lying on these fish here. Seemingly, yes, innocuous little thing, but a sea lice that costs the industry around probably between 5 and 10 billion Norwegian krone every year. So that is probably enemy number 1 for Atlantic salmon farming, the sea lice. So just a quick overview of the history of the Marine Harvest breeding program. So it started in the 60s from basically eggs from 2 river systems in Norway. For a significant period of time, there was just mass selection, very basic, with control of inbreeding. In 1999, this was changed to a family-based breeding program, which enabled, of course, a lot more sophistication in the breeding program work. And then we introduced QTL and genomic selection really from about 2012 onwards. And I think I'd like to say now from 2017, we've really transitioned to a full genomics in the breeding program. And so why genomics? Well, we're animal breeders here, we're hearing a lot about, of course, genomics these days. I don't need to say too much about why genomics. Obviously, the increase in accuracy of breeding values is one of the most obvious results we can get from genomics in a breeding program. Donna mentioned, of course, some of the other advantages, of course, of using genomics, some of the other effects, the byproducts in a way, and the other things that it benefits the breeding program. But this is just a way to show, well, traditionally, if we had a 40 individuals from a single full sibling family in a salmon family. If we had just a family breeding value, all those individuals would have the same breeding value. We don't have an individual breeding value on those because the family breeding value is all that exists. However, if we look at the genomic level, we had an individual genomic breeding value for all these individuals, we see that, of course, there's a big range within that family. So not only does this mean that by using genomics we can select the best fish within the best families, we can also select a lot of good fish from families that would otherwise have had an average that was not particularly good. So the Atlantic salmon genome was sequenced in 2015, published at that time. It was a significant and important resource both for the research community and for the breeding companies, giving access, of course, to 3 billion bases of sequence, around 46,000 genes, and, well, how many million SNPs? Probably tens of millions of SNPs. This is a work in progress. This figure here shows a little bit of the complication of the salmon genome being ancestrally duplicated genome that is basically reverted back to diploidy. However, effectively, each chromosome is represented, each chromosome is duplicated in the genome. So you can see here where these straps are joining, that's effectively the same sequence. And that creates some challenges for, of course, for SNP genotyping. I won't talk too much about that, but it's a complication that we have to deal with Atlantic salmon genomics. So a little bit about our use of Axiom arrays in the breeding program. We started off in around 2013 with the first Axiom array, which we call NOFCell, collaboration between the Research Institute in FEMA and another breeding company, Salmon. That was a 37K product, and we genotyped all together about 5,000 samples. So it was a small scale. It was a discovery, basically a discovery project of R&D, with the main focus on R&D. We followed up 2015 with the second version, where we pushed it up to a 57K array. And then we also together scaled that up to 20,000 samples. So we moved, one step further in the process. And from 2017, we had our third array, which was the NOFCell 3 array, another 57K array. But the big difference here is that together we've scaled things up to 225,000 samples in a commitment that we've made in that purchase. The obvious, we've heard a little bit about, of course, economy of scale is a very important thing here. We pay a lot less when we order 225,000 samples compared to 5,000 samples. So there's a significant benefit in upscaling from the cost side as well. So how did we design these arrays? I won't spend too much time on this because I can see my clock is ticking down there. But basically, the original NOFCell array, we didn't have a salmon genome assembled in chromosomes. So we basically mined existing RNA-seq data that we had and selected what we thought were a polymorphic and informative SNPs focused more on coding regions, if possible. So we used some typical parameters like minor allele frequency, coverage, and putative function to help us down select that set. We ended up with around 30K polymorphic high-resolution SNPs, and that was a good starting point for us. We generated a lot of data from that, and it was a good foundation for the next version. So what we did here was we took the good SNPs from the previous array. We now had a genome assembly assembled in chromosomes, so we mapped these SNPs across the chromosomes, and we filled the empty spots on the chromosomes with SNPs from the existing, from the public Axiom salmon array, which is a commercial product offered by Thermo. So just to visualise that, we had SNPs. The orange SNPs here is the ones that we were able to map to the genome, and then we filled in the gaps where we could with other polymorphic SNPs. Then we went to NOFCell 3, pretty much the same process. We took the good SNPs from NOFCell 2. We found some additional other targeted SNPs that we generated through various R&D projects. So we added those to the array. You heard from Donna how we can, of course, refine these arrays. So we just added a new set of SNPs. We genotyped 2 more of the commercial salmon array. And again, we just did that same process of refinement of filling in bins. So here we had 2 SNPs that didn't perform as well as we hoped. So we got rid of those and then just replaced them with 2 new good SNPs. So by the 3rd iteration of this array, we're sort of getting to the point where we've got a really solid set of SNPs, and it's very informative. So how is our analysis pipeline? Well, we have a lab we work with, a project tracing system. We send them samples. They extract and genotype and send us results. They do all the plate level QC work at the lab there, and then they upload the raw data in terms of cell files to their server, and we download and do the following analysis. We have a pretty efficient pipeline now for this. I don't think I'll go into the details here, but it's an APT pipeline on a Unix server, and it does all the clustering, genotype calling, and we also have what we call a rescue step for basically recovering the good data from samples that just fall below that threshold. If we set a threshold of 97% and we have a sample that has 96.9% core rate, if we were very strict, we'd lose that sample altogether, but we know there's a whole lot of good data in that sample, so we have this rescue step where we get the good data from that sample. And then we end up with producing some binary plink files as an output. Then we take that data and we do the various things. Analysis suites, of course, we run the usual things, sample QC, SNP QC, we apply our marker maps to the data, and then we're at the applied level now for the breeding program where we're doing GBV predictions, and we're doing GWAS, and, well, as you know, anyone who's worked with large volume SNP data, there's an awful lot of stuff you can do within this data in addition. So it's a huge resource, and we've generated, 60, 70, 80,000 samples now in our data sets, so they're very, very significant. This pipeline is very fast now. We've got this down to the point where we can go from raw data to GBVs in a day, basically. So I can download the cell files, run the pipeline, do gblup, and have this data in a single day. And why it needs to be that fast? Well, it doesn't always need to be that fast, but sometimes if we need a quick turnaround, if we have some fish that we have sampled and we need to get them on land at a certain time and pre-select them, then we need to do this quickly. And so this works. It works really well. Genomic selection, standard methods. We do it the same way as livestock breeders. We have a reference population, for example, disease challenge test. We send a group of fish to a challenge, they get exposed to a disease, and we record the phenotype, dead, alive, or some other quantitative phenotype. We genotype them with the Axiom array. We have our prediction equations. I mentioned gblup. We also look at Bayesian models. We find that gblup works really well, and we don't get a lot of additional benefit from the more complex models. Maybe that's something with salmon population structure in the families that we have. We have our candidates, and of course we genotype them and have a breeding value, genome breeding value. So this is not hugely different really from any other livestock genomic selection. And it really works for us. I mean, we've looked at this. We've got now we've got, of course, cross-validation. We've got some real validation data, but we get a really significant, substantial increase in breeding value accuracy across the board. I apologise, some Norwegian words have snuck in here, but this is a viral disease. That's a viral disease. That's this sea lice, fillet colour, and growth and weight. And you see these are significant increases in accuracy. And we see that across the board. So, we're really seeing an effective result coming out of this genotyping work now. And we're also finding QTLs. Now, if any of you were in the aquaculture session yesterday, you would have heard from Aquagen how, they've spent a lot of resources and time, discovered significant QTLs, major QTLs, and have selected on these QTLs for a period of time and are seeing really good results. We're finding the same thing in our data, in our population. This is an example of a major QTL for a viral disease. Obviously, those who are familiar with Manhattan plots, a major effect here on one chromosome. Huge difference between genotypes in mortality and survival. And, this is a trait that has been implemented for some years in the industry and has had a really positive effect on the number of outbreaks of that disease. Another QTL, another viral disease for us. Again, hard to see. Sorry about that. Manhattan plots are always horrible to show, I think, on screens. But at the end, we've got a major QTL here, another one here, and when you look at the fish with 4 copies of the really weak allele, they had 94% mortality, and the fish that had 4 copies that are resistant allele at these 2 QTLs had only 25% mortality. So really significant effects here we're finding in these QTLs. I can see it's flashing at me now, so I'm going to move on fairly quickly. We've also used this to verify what's called the puberty gene in salmon. This is a locus that controls basically age at maturity. Fish that mature after one sea winter, one mutation effectively controls this. And this was discovered in wild populations and published in Nature, and we've also shown this in our breeding population. And this has some implications for the breeding program in terms of being able to screen out fish that will mature too early for us. Sex determination, there is a gene that controls sex in determination in salmon called STY. We also talked about in the aquaculture session yesterday. Simple and accurate sex determination, we have these probe sets on the array. Basically, when you detect the gene here, it's a male. When you don't detect it here, it's a female. So we draw this line here. These are male, these are females. This is really useful data on small immature fish that we can't sex and we want to, for example, split into males and females. It's not 100%, and it's not because of the array. That's because of some weirdness in the salmon genome, which some of you know about. But it's a very useful tool, and it's included in the array, of course. So we get it as a bonus in a way for all the genotyping. So just to conclude, then, I think this large-scale implementation of Axiom genotyping have transformed our breeding program. We went from this in a sort of an R&D perspective, looking at it, testing it, a few studies, then seeing that it worked, and then basically deciding we'll go all in on this. And now basically all our breeding program revolves around genomics, and all our breeding values are genomic breeding values. We see really a good improvement in accuracy from genomic selection now. I think all the salmon farmers, all the salmon breeding companies now working with this get equally good results, so it's really positive. We're finding major QTL, another really useful thing for us because we can do some targeted genotyping and select on QTLs. And the Axiom arrays, they're good. I mean, it works. It's high throughput. We get very reproducible results. It's fast. Like I said, we can go from raw data to breeding values in a day. And that's what we need. We need something that is reliable. We have large numbers of samples coming from different places, different times. They need to go through a very standard pipeline and get reliable data. And our software and analysis pipelines, I think, have reached a good state of maturity and efficiency, and we get that high throughput and speed, which it's actually quite important for us. So, thank you very much for your attention.